So our first presenter is Carly Charron, a fourth-year PhD candidate in biology and is supervised by Rima Manassa. Carly obtained her undergraduate degree in genetics at Western in 2020. She's now a PhD student in biology and works at Agriculture and Agri-Food Canada. Her research is focused on using plants to produce an edible vaccine to prevent Salmonella colonization in poultry. She's passionate about outreach activities at Western, including the GradCast, Let's Talk Science, and Science Rendezvous. So please, welcome Carly.
So most of you have probably heard of the bacteria Salmonella. It's one of the leading causes of foodborne illness and results in unpleasant symptoms such as nausea, vomiting, and diarrhea. And while for most individuals these symptoms are relatively mild and short-lived, hospitalizations and deaths can occur, which poses a significant concern for public health. Salmonella poses economic concerns as well, as there are large costs associated with Salmonella contamination, as well as with the treatment of infected individuals. So we can contract Salmonella from a variety of food sources, with the most common being poultry products. And poultry pose a major concern as they can harbor large loads of Salmonella without displaying any symptoms, allowing the bacteria to easily spread through entire poultry flocks. This can also lead to the contamination of food crops, such as fruits and vegetables, through the use of contaminated chicken manure in fertilizer. So because of these issues, there is a really big need to develop effective control measures to prevent Salmonella colonization in poultry. So historically, antibiotics have been one of the most commonly used measures. However, their use is being limited with the rise of antibiotic-resistant bacteria. Vaccination is now one of the most common measures. However, the vaccines that are currently available either pose safety concerns or have low efficacy and are often expensive for farmers as they require administration by a veterinarian. So because of this, the goal of my project is to develop an edible plant-based Salmonella vaccine that would be safer, more effective, and cheaper than those currently available. And plant-based vaccines have many benefits, as plants are easy and inexpensive to grow, and the leaves of the plant that actually contain the vaccine can be dried and ground up into a powder that can be directly fed to animals. And this would significantly reduce costs for farmers, as it would eliminate the need for administration by a veterinarian. So for my project, I've selected a protein from Salmonella to use as an antigen for my vaccine. And from this protein, I specifically selected these small extracellular immunogenic regions and genetically fused them to a protein nanoparticle, as seen here. So these protein nanoparticles are very large, dense, and globular proteins that are very easily recognized by the immune system and can induce a very strong immune response. So I produced very large quantities of these proteins in the plant N-benthamiana, which is a relative of tobacco. And when these proteins were administered to mice via injection, a very strong immune response was induced, confirming that these proteins are in fact immunogenic. So my next step will be to perform a vaccine trial in chickens, where ground up leaf powder will be fed directly to one-day-old chicks. And if this is successful, this could lead to the development of an effective vaccine that would prevent Salmonella colonization in poultry and subsequently limit the risk of foodborne illness in humans. Thank you.